Hello, my name is Elizabeth Whitson, and I am a second-year master's student here at the University of Miami's Rosenstiel School for Marine and Atmospheric Science. I am conducting my research in the Marine Microbial and Evolution Lab with Dr. Javier Del Campo.
My research is focused on the sea hare Aplysia. Here at Rasmus, our lab has been able to collaborate with the National Research for Aplysia Laboratory, which is part of Rasmus's experimental hatchery. Aplysia, or their full scientific name, Aplysia californica, is a well-studied marine mollusk used primarily for neurological studies. So Aplysia are actually native to the eastern Pacific Ocean, and as their name probably suggests, they can be found off the California coast. They can also be found in northwestern Mexico and in the Gulf of California. So Aplysia can be found in shallow water ecosystems, primarily the intertidal zones. They primarily eat algae as their main food source, and they prefer red macroalgae. They actually get their color from their food, as we can see, and depending on what type of algae they're eating, it can actually change their body colors. This ability to match the color of their food provides really good camouflage in their ecosystems. So they're able to hide really well from potential predators. Aplysia are also hermaphrodites, which means that they have both male and female sex parts. All of this information is really important for the hatchery here at Rasmus, since they rear hundreds to thousands of Aplysia a year for research purposes.
Aplysia have simple neurological systems with well-defined and large neurons, a relatively short life cycle, so about a year long, and they live in a semi-controlled environment at the hatchery. So all of this plus more is what makes them great models for these kinds of studies. There have been various studies conducted over the past few decades on Aplysia that look into topics such as behavioral and neurological changes with age, hypoxia, their life history, etc. So their genome and transcriptome have actually been sequenced. However, their microbiome has not, and this is where my research begins. For those who are unfamiliar with some of the terms I have thrown out, let me define them. I first talked about how Aplysia make great model organisms, but what exactly is a model organism? A model organism is essentially a non-human species that are used in laboratory settings to help scientists better understand biological processes. I also brought up terms such as genome, transcriptome, and microbiome. A genome is essentially the complete or the most complete version of all the genes present in a species or organism. A transcriptome follows the same logic. However, instead of genes, it is instead RNA in a particular tissue, cell, or organism. Lastly, the microbiome, which is what my research is focused on, is the collection of all the microbial organisms and their metagenomes in a predetermined host species or organism. A metagenome, as you might guess, follows a similar definition to genome, but it is inclusive of more than one organism or species. Microbial organisms include things such as bacteria, archaea, protists, viruses, and fungi. So I am specifically focusing on bacteria and prokaryotes and possibly microeukaryotes for my research. Now I'm going to change gears a little bit and talk about another aspect of my thesis work. So in the past 15 years, there has been a large emergence of studies being done on microbiomes and how they are tied to neurological diseases, psychological disorders, and health. Many studies have highlighted the importance of the composition of gut bacteria and its connection to the gut-brain axis. For the purpose of this video, I'm not going to go too complicated or too in-depth, but essentially the gut-brain axis is a bidirectional communication and signaling system between the gut and the brain that is done through many different biological processes. The system is bidirectional, or goes both ways, meaning that both the brain and the gut is hypothesized to have influence over one another. A big component to consider here is bacterial gut composition, since in the human gut alone, there are estimated to be around 100 trillion microorganisms. most of which are bacteria. The human gut has the highest density and the highest abundance of microbes in the human body. So gut composition has also been linked to neurological diseases and psychological disorders, including anxiety, depression, stress, Alzheimer's disease, Parkinson's disease, multiple sclerosis, autism spectrum disorder, and schizophrenia. So, bringing all these ideas together, we aim to alter the microbiome of this model species that is highly used for neurological studies. So, thank you all for watching. If you have any questions, you can email me at exw195@miami.edu. And thanks again for watching.